My name is Wojciech Rosikiewicz, and I wanted to show you one of the use cases of our CovidExpress portal. And in the paper describing this portal, we refer to this use example as case 2, study case 2. And the rationale is that we can explore all those data that we have reanalyzed, all COVID-19 related data sets that we have reanalyzed, and we can explore them, for example, using the list of genes of interest from the users. So for the purpose of this exercise, we start from a KEGG database where there is a gene set collection of the pathways relevant for COVID-19 disease. So as you can see, let me reload that, there are multiple entries, there are many genes and multiple pathways within that one pathway. There is actually, when we look into details, there are over 230 genes listed for COVID-19 disease. So we don't want to go, and in the paper also, we didn't want to go for the full collection. It's relatively high. I don't expect all of them or go up or down in response to COVID-19 infection. So rather to narrow this down to a smaller set of genes, we used EnrichR, a very good tool for pathway enrichment analysis. We took those 230 genes and, as we described in the paper, we reviewed those results, and what caught our attention was actually from ontologies within MGI phenotype level 4 database. There was this one specifically, increased susceptibility to bacterial infection, which caught our attention. So when we downloaded this database, those results from EnrichR for this database, then we actually end up with the list of 26, if I'm correct, 26 genes, which are those genes. I already had them in advance for you. They include ACE2, among others. False, very important. So with this list of genes, we then wanted to ask, are they upregulated? We know that they are important for COVID-19 disease, and we know that they are enriched with or associated with increased susceptibility to bacterial infection. So what happens, the question that we wanted to ask, what happens with them when different phenotypes from COVID versus control, for example, are explored? So for that purpose, we can actually, with those 26 genes, now use our CovidExpress web portal. So for this, we simply go into our website and then open the server. I already did that. Here we can just put any kind of name. It's not that important actually. We create an annotation file, and then we can start explore. So in this, like I mentioned, in this use case, we are going to explore the GSEA analysis part. So we click here. We don't want to use those example genes, so we just select them and remove. Instead, we put the list of those 26 genes that we just got from EnrichR, and then we just click to analyze those. So by default, it would display top 10 GSEA results, and it will use fold change rank. These are the default settings, but other are available as well. This takes time. It's actually a lot of calculations to be done in the background. So like it says, 30 to maybe 60 seconds is needed for this. And maybe I can already also tell you that those results that we are going to see are not going to be exactly the same as those that we described in the paper, and that is because the GSEA is using bootstrapping, and because of that, those normalized enrichment scores that we report are going to be slightly different every single run when we repeat the analysis, even with the same input genes. So there is going to be some variability, but if the signal is, for example, let's say those genes would be upregulated, they would always, from what we observed through using GSEA not only here but in research every day, those strong signatures are always there, and we are going to show top 10. So those usually those could be different top 10, but still they would have the same direction. So yes, already we can see this summary of top 10 enriched contrasts, and we can see that those 26 genes related with an increased susceptibility to bacterial infection, we can see in which contrasts those genes were up or down regulated, up or down. So essentially they were enriched here with negative NES score toward control phenotype in this case. And here, for example, they were enriched toward treatment, so the positive phenotype. So we can view now that, for example, we can see that they are enriched, so upregulated in COVID versus control samples, in CALU cells, in organoids, in some corneal cell, and yeah, CALU to COVIDs, like 24 versus 4 hours is the control. So the longer it goes since infection, the higher the expression of those genes. So let's just explore that further. In the meantime, as you can see, H1299, this is different cell type, cell line, sorry. So we can see that CALU and H1299 behave kind of opposite way for the time since infection. So once we click on any of those, then we actually can view the results. We can download the GSEA enrichment results. We can also download those results. Yes, we can view those genes, enrichment plots. We can save that and directly use in the publication. So yes, overall this approach shown here has the power to quickly narrow down your search to specific studies that show or support up or down regulation of those genes of interest. And I believe that's very useful. Okay.